All right, let's talk about blood types.
Blood consists of several different types of cells, including red blood cells and white blood cells.
Today we'll be focusing on red blood cells, also called erythrocytes, and how these cells determine what blood type you are. Let's look at an erythrocyte. Located on the surface of the cell are important protein receptors called antigens. These antigens come in three primary types: A, B, and Rh, sometimes called D, but we'll call it Rh. Now let's look at the blood plasma. Located in the blood plasma are some Y-shaped proteins that are called antibodies. There are hundreds of different types of antibodies in your blood, but for now we'll only focus on the A, B, and Rh antibodies. These antibodies are produced by the immune system and are used to identify and neutralize foreign objects in the body. Each antibody is specific to an antigen, meaning one specific type of antibody will only recognize one specific type of antigen. An A-type antibody is rigged to recognize A-type antigens, and B-type antibodies recognize and tag cells with B-type antigens for destruction. The differences in human blood are due to the presence or absence of these antigens and antibodies.
So when we're talking about what blood type a person has, we're referring to what antigens are found on their erythrocytes. There are four main blood types in humans: A, B, AB, and O. A person with type A blood will have A antigens on their red blood cells and B antibodies in their plasma. B blood types will have B antigens on their cells and A antibodies in their plasma. A person with AB blood will have both A and B antigens on their cells and no antibodies in their blood. If they did, their immune system would mount a response against their own blood. Just think of it all as a big BYU party. If someone shows up to the party wearing a red shirt for the University of Utah, the party bouncers are going to throw him out. It's the same way with the party in our bloodstream. If a cell enters our bloodstream with a different antigen than what the other cells have, the antibodies are trained to search out and destroy that cell.
Finally, a person with type O blood has no antigens on their cells, but has both A and B antibodies in their plasma. Type O is sometimes called the universal donor. Why do you think that is? Because their cells have no antigens on them, they won't be recognized as foreign no matter what types of antibodies the receiver has. This is the partygoer that has neither a Utah or BYU shirt on. She won't get thrown out of anyone's party.
Let's move on to discuss the Rh factor.
This is where blood typing can get tricky, so pay close attention.
Rh is an antigen, just like A and B, and as such, it has an antibody specific to it called the Rh antibody. If a person has Rh antigens on their red blood cells, we call them positive. If they don't, we call them negative. Let's say this blood cell has Rh and A-type antigens on it. What blood type does this person have? A positive. What type of antibodies do they have in their plasma? B antibodies. We don't want A or Rh antibodies in this instance, or they would initiate an immune response against the blood. To summarize this concept, we call a person with Rh antigens positive and a person without Rh antigens negative. When I'm typing blood, I do it in two steps. First, I figure out which of the four blood types it is, A, B, AB, or O, by finding out what antigens are found on the blood cells. Once that's done, I figure out if they're positive or negative by finding if their blood cells have Rh antigens on them or not. To experimentally find somebody's blood type, we use three different types of synthetic antibodies: anti-A, B, and Rh serum. A person pricks their finger and mixes a drop of their blood with each of the three serums. The fourth drop will be the control. We then observe what happens. When blood cells carrying one or more of the antigens are exposed to the corresponding antibody, an immune reaction occurs and they agglutinate, meaning they stick together and form clumps. Thus, we can identify the blood type by noticing the agglutination on the corresponding drops.